p-adjusted value less than 0.05. I think then you can get important pathways. Yeah.
Okay.
Okay, so now I would ask Ilya and Dr. Raghavendran to give comments on your presentation or any suggestion that they have.
Yeah, thank you. I think overall, it's clear that you've done a lot of work, and now the task ahead is to integrate all of these analysis results and kind of make sense of what are the clear findings. I think because of the time constraints, this is an exciting update to the project that you've been working on for a while, and you're obviously addressing a lot of the issues that came up during the initial analysis. Let's move on to hear a little bit from Dr. Raghav, who is another mentor, and then he can transition to his part of the presentation. So thank you, Urja.
Thank you, Ilya. I think I'm audible. So that was a very detailed and very good work, Urja. And I'll be interested to see. Okay, it is not there. Yes. Okay, great. So I'm very much interested to see the pathway associated with this, and then you can do selected analysis on the pathway that are shared or that are not shared, completely different between the normal stem cells and the glioma stem cells. So those are very interesting ones. Maybe an even more stringent p-value could give you a reduced set of genes, because you still have a lot of genes to analyze, and that would, yeah. So you can maybe you can reduce your analysis or downstream analysis to top 20 or top 15 upregulated genes and top 20 downregulated genes for each set, and then try to see if these top 20 genes are from multiple pathways or the pathways associated with these top 20 genes. That should narrow it down and make your analysis even more workable, because I mean, at the moment you have really a lot of data to comprehend with. So right now, I would start with that, and then and see where it goes, and maybe extend it to top 50 if that makes even more meaningful, even more meaning, and we will see. Yeah.
All right. Thank
okay. So thank you. Cheers. So I think that will translate or transition to my talk or presentation. I will share my screen right away. I just need to find it where. Okay, got it. Okay, excellent. Okay, that worked. Okay, great. Can everybody see my screen? I think you can. Yeah.
Yes, sir, we can. Thank you.
So yeah, thank you. I'm Raghavendran, bioinformatics mentor and research consultant with Cyomics Logic. Let me brief you about my background and research interests. So I grew up to be a tinkerer kid who is interested in solving many problems. So right in my younger ages, computers and programming with computers introduced me to very new puzzles and challenges. So attempting to solve these puzzles and these challenges were the first step that took me towards solving puzzles and asking questions. So during my undergrad education, I read about the potential and the announcement of the Human Genome Project. And that was the first research article that I read. I didn't understand it, most of it, but it inspired me to a great deal, and I pursued my master's in biophysics. So my natural inclination towards computers and structural biology, bioinformatics, led me to specialization in computational biology and bioinformatics. So after my master's in bioinformatics or biophysics, I joined a PhD in Max Planck Institute for biophysical chemistry with a specialization in computational biology. So during this tenure, I developed software and designed a lot of methodologies and pipelines for protein structural and dynamic studies. So my system of interest during the PhD period was proteins associated with neurodegenerative diseases. So when at the time when the next-gen sequencing technologies were starting to get established in India, I came back to India as a postdoc to the Institute of Bioinformatics and Applied Biotechnology in Bengaluru. The potential of the next-gen sequencing technique and its application to multi-omics data analysis, these two combined and piqued my interest in cancer. So you all know cancer is the leading cause of disease death worldwide. So I got naturally inspired and interested to work with cancer biology. So I contacted Professor Shandar Ahmed in JNU and Dr. Pedro Ballester in collaboration with Dr. Pedro Ballester in INSERM, France. I proposed a study to understand and to explore tumor responses to different treatment regimes. So this was while I was in Bengaluru, and I also agreed to help a friend who was in need of bioinformatics help in de novo genome assembly of a variety of neem plant. So it was only during this collaborative project I came to realize how little information was available for plant bioinformatics in the public repositories. While when you take or when you compare it with the availability of databases and the repositories for human or bacteria, there is hardly anything for the plants. But there is so much potential to understand plant genetics, because through bioinformatics and through sequencing techniques, we can understand the underlying biology of plant genetics. Because agricultural crops have been subjected to artificial selection for ages, irrespective of geographical location and irrespective of culture. So I continue to work with that project, and they are writing up the paper, and I'm still associated with the department for some troubleshooting in the pipeline that I developed. So while all this was going on, the project that we proposed was successfully funded, and so I joined JNU as a postdoc research scientist. So I will detail more about the data that I use for projects and about the projects and the results in a short. But I just wanted to mention, right from my PhD years, I enjoyed working with undergrad students and enjoyed working with students. So this experience tremendously inspires me, and I feel privileged to work with some of the bright young minds. So I hope and I wish every one of you will enjoy working with me, and as much as I look forward to working with you. So for the next two slides, I want to move on to speak about the data that I use for the project on precision oncology. Let's see if I can get this rolling. Yes, okay, great. So every once in a while in science, a groundbreaking research gets published, and the data collected for that work contains a lot of potential to answer a wide variety of scientific questions and queries. So this is one such paper. And this paper is about tumor cells that are grafted into mouse models to study tumor responses to different treatment regimes. So what they did was they extracted the tumor cells from cancer patients, and then they sequenced it. And from the sequence, they profiled the gene expression levels and the gene mutation levels and the copy number variation levels. And then these tumor cells are grafted into immunosuppressant mouse. So, and these mouse were administered different set of treatment regimes, and the tumor's response to these treatment regimes were studied. And such mouse or such immunosuppressant mouse which got administered with tumor grafts are known as patient-derived xenografts. So these PDXs, in short, and they are administered 60 different treatments, and their response were recorded in change in volume and the ability of the mouse to survive the disease. So such survival data analysis of 1,075 PDX mouse was released along with this data, along with the study. So this study from this analysis concludes that the mouse models provide a better alternative compared to the cell lines which were traditionally used, because cell lines are often affected by the petri dishes they are grown on, and heterogeneity is one of the biggest problems in cell line data so far. And at the basic genomic level, compared to the cell lines, the genes, the gene expression levels of the PDXs resemble more to the cancer patients than the cell line data. And PDXs are also consistent, or the data from these PDXs are also consistent with extensively studied therapies, and this comparison validates their applicability to understand the underlying cancer biology. So such a wealthy of data generates a plethora of research questions, as I already posed. So one can employ this data to various analytical pipelines to study treatment stratification, like can the mutations in genomic data predict tumor resistance, or vice versa? Can tumor resistance relate to significant mutations in those models? Or are combination of drugs more effective than single drugs? Or how does different combinations of drug work well or does not work well together? Or can we find the underlying biology behind this? Or we can also do some precision medicine study, like do targeted therapy exhibit higher efficacy than general cytotoxic treatments? Because among these 60 treatments, some were general cytotoxic treatments, which are toxic to all cells, just they kill cancer cells more than the normal cells. And some specifically target pathways inside the cancer cells. So can we find out the underlying differences between these two treatments? And with the help of multi-omics integration, can the PDXs broadly represent the interpatient diversity? Because that was one of the biggest conclusion of this study, where they observed a diversity among the models which carry the same cancer cells from different patients. So they translate into interpatient diversity. So can we quantify this diversity? Or more importantly, can we find the commonality among this diversity so that we can understand or we can use it for biomarker analysis? So these are different questions. And what I used this study to work on. So my aim was to use this PDX data to design and implement machine learning models to predict tumor responses to different treatment regimes. And why do we need to do that? So the efficacy or the effectiveness of the cancer drugs is very strongly patient dependent. That is why we are moving more and more towards precision oncology and personalized medicine, because on an average, only 25% of the cancer patients actually respond to cancer drugs. But unlike your normal painkillers, you will have 80% of the patients responding positively to the painkillers. So currently there is a need to predict which patient will respond positively to the treatment. And if not, there is no need to spend time and energy to treat the patients with the treatment. So for this study, one obviously cannot use patient data because of the difficulty in the uniformity among the patient data, but we can actually use these mouse model data to predict the treatment response. So how did I do that? The gene expression data, the copy number variation data, and the gene mutation data were the features from which the machine learning models defines a function to understand the tumor response. So when this model was presented a test case, it tries to predict the tumor response to that particular drug. So such a study, this model was applied to breast cancer and colorectal cancer PDXs, which were treated for 26 different treatments. So what did we find? Traditionally, canonically, single genes were used as biomarkers that predict the onset of cancer. And the same set of single genes were used as a treatment biomarker also. For example, if there is an association of a gene to the onset of cancer, the same gene was studied whether this particular cancer will respond positively or negatively to any given treatment. This is obviously and logically is not a very accurate predictor. So this is not a very successful model, but from a multi-omic data analysis, what we demonstrate is before that I want to introduce you to the accuracy measure that we are measuring the machine learning models accuracy levels. So this is Matthew's correlation coefficient, where an accuracy of 1 would mean the perfect accuracy, like all the test data are perfectly identified or predicted to be how it responded to the treatment. And -1 would be complete opposite. If there is a positive response, the model predicts a negative response. And around 0 would be a random response. So what we want is these data points to move more towards 1 so that we get better accuracy of our machine learning model. So in that one, so when the machine learning model was used to predict on single gene markers, you see the accuracy is not very great. It's actually, it's very bad. So when multi-gene models were introduced in place of single gene models, the accuracy is improved. And the accuracy level can further be improved by combining or by including more than one profiles or more than one molecular profile, like gene mutation data combined with gene expression data, combined with copy number variation data, improves accuracy tremendously. So to quantify, we actually observed, we could predict 19 out of 26 treatment regimes. We could predict correctly 19 out of 26 treatment regimes. So this was what we performed, and what could be done, what could be done more in such a study. So one can extend such an analysis to include patient data from TCGA repository, but that involves extensive curation of the data, but one can still do it. A gene set enrichment analysis, like what Oja presented, of the biomarker genes will throw insight into the biology behind the response of these tumors to different treatment levels. So this is how we did. And such a study was actually performed for selected cancer and for selected treatments. So one can obviously extend the study to all the cancer to find the commonality of these cancer cells to react to different treatment regimes. So these are the different future steps that the team is currently working on, but I finished my part and I joined Cyomics Logics. So I hope this project was informative and it does inspire you to utilize curated PDX data. This curated PDX data will be available at the Cyomics Logics platform shortly. And this, this platform, as you are aware by now, with different introduction from Elia and from Harpreet and from Sonalika, provides a very well-organized research fellowship program that I want to speak about in the next 2 slides. Let me go to the next slide. So yes, so from my research experience, there are 3 main challenges that all researchers will need to overcome in any research program. And this research program is designed to target specifically to address these. 1, time. So a lot of time is actually spent on learning research methods from multiple sources with a varying level of reliability. But in the end, you are going to use only 1 or 2 methods for your question of interest. So what we have, the learning platform, the Cyomics Logic learning platform, provides you with a very well-planned learning modules and coursework. This coursework will prepare you for any challenges you might face with the data analysis and or the biological interpretation of your data analysis and your research question. And another big challenge in doing computational work or bioinformatics work is the presence of, or is rather, there is a limited access to bioinformatics expertise or a reduced availability to bioinformatics experts for the students. So in this program, the research fellows will participate in cutting-edge bioinformatics research led by mentors like me, like Harpreet, and several other mentors. You will find information in the website. We have years of expertise in computational biology, bioinformatics, and also including emerging domains like spaceomics. So this network of mentors that we have will provide you with expert feedback and peer review from the inception of your topic to the final results. And these results will be worthy to be sharing among the scientific community and to the world. And one final challenge is limited access to infrastructure that is required to perform big data analysis. I think this is the most important challenge that many students face right now. So the cloud-based analytical Tauber bioinformatics platform that you are introduced to today, it provides all the infrastructure that is necessary to perform big data analysis in an intuitive environment with the help of flexible, interactive, and visual pipelines. So the research fellows will get access to this platform and a list of curated public data sets with which you can design a big data analytics pipeline on multi-omics data, or one can perform translation research, or one can address clinical questions, or biomarker identification, or treatment stratification, or variety of questions that you can think about leveraging this data and infrastructure. So who can benefit from such a research fellowship program? So this research fellowship program has been designed to help young research and students like you to gain access to the premium resources on bioinformatics for the analysis of complex high throughput data sets that I presented before. And like Dr. Harpreet also presented, and also we observed from OJA also. So, and the experts will help you complete a research study on your own. So if you are an undergraduate or a master's student, you can actually engage in research and you can develop a deeper understanding of the subject matter than that is possible through your classroom routines or classroom instructions alone. So such an early exposure to research has been proven to be very helpful and can be the perfect launchpad for your career. You can also apply the skills. That is one of the most important points. You can apply all the skills practically, the skills that you learned from your classroom instructions, to a project of your own and to the project of your that piques your interest. And more documentation wise, you can fulfill the requirements set by your university syllabus or curriculum. So if you are a research student, so we provide, you'll get an excellent opportunity to work one-on-one with the network of mentors and to use the TBio big data infrastructure to be part of your research question, or to be an independent study to develop new skills that you are interested to do that. So with that, I thank you all for your interest and attention. And this concludes my presentation. And I think I would like to pass the stage to Subrahmanyam Barua. He's an astrobiologist and he works on space omics and space health management. It's very exciting to hear from you, Subrahmanyam. Please take over the stage.
Well, thank you so much, Dr. Raghav, and it was lovely hearing to you and your perspective towards bioinformatics, its usage. It was amazing to learn about the...
Sorry, any questions? Yeah, please go ahead.
Sure. So it was amazing to learn, and I hope that our audience also has enjoyed every bit of it. And I hope that everybody is as excited as I am for this particular research opportunity, which we call the research fellowship. Now, quickly, I would just talk about a student club, which is called Cyomics, and what is the agenda of this club, what are we targeting to do, and what would you benefit by being a part of it? Well, hello everyone, and thanks for tuning in to today's webinar. It was amazing to see you all here, meet you virtually, and also learn about such amazing opportunities and research going around. And I'm sure that you are also kind of interested or already starting to hope that what can you do and how can you contribute and how can you also be a part of this exciting community. Well, that's something that I am going to talk about today. So my name is Subrahmanyam Barua, and I am the Scientific Outreach Coordinator at Fine Biotech, and I'm also the president of the Cyomics Student Club. I have an MS in Molecular Biology and Biotechnology from ITMA University, St. Petersburg, Russia, and I am a researcher at the International Laboratory of Solutions Chemistry and Applied Technologies. Well, to begin with, Cyomics have been making an impact all throughout the globe since its inception. We have a huge community of more than 1,000 enthusiasts all over the world. We have been exposed to more than 20 great scientists and their webinars all throughout. We also have a very strong alumni network that consists of more than 30 successful candidates from all over the world who have been a part of Omics Logic Research Fellowship or Fine Biotech's research internships, and now they are out in the world making an impact, making a difference all throughout the globe. So let me talk a little bit about what Cyomics is all about. And to do that, I would begin with a very simple story of every student, because, you know, I have been a student, I am a student even now. So I pretty well recognize every student going through something of this sort. Well, I know that you guys would also agree. And if you can agree to that, do let me know in the chat section that we do get interested in doing a research very often, right? We get some idea, we get to know about some information, we watch some webinars, some presentations, and we are so hyped up to begin our own research journey. Maybe we have a task to do, we have an internship to do, or a project to submit as a part of our college curriculum. So we do get interested in doing a research, but there are certain barriers because of which we are unable to complete it, or even if we complete it, then we are not very satisfied with whatever we get, and we just hope that there would have been something better that we could have done. So what are these barriers? Well, sometimes we are unable to define a proper research question which is of relevance and which would be appreciated by the scientific community, because the scientific community is so dynamic. It keeps on changing every day. So maybe we are not able to define the question as required. Sometimes we face one of the major hurdles while doing any research project is we can't find a good data set, just like Dr. Raghava and also Ilya earlier was talking about, that finding a good data set which is curated and which is worthy of analysis bioinformatically using the various tools and algorithms statistically is a major challenge and a very difficult task. Finally, there can also arise a lack of skill sets which are required in this modern world. Well, we might know a lot of skills, we might have experience in many things, but the world changes so fast. So maybe we need to develop and increase the level of expertise that we have in the current technologies out there in the world. But why a student can't address these issues? Well, there can be various reasons. Some of the possible reasons might be there's no one to guide this particular student. They don't find someone enough close to them who they can express their problem, their situation, and who can be like a mentor to them. Then maybe they have some supervisors in their college or their laboratories, but they're too scared or shy to talk to them and consult them. And maybe they often think that the question that they have or the doubt that they are bearing in their mind is too silly to ask someone. They might not know the places where you can find good quality data for your analysis out there, which are publicly available for anyone to analyze, but we don't really have the knowledge of where to look for the data. Finally, there can be a major issue of lack of motivation. We might have that enthusiasm to begin with a project, but maybe in the long run, we just lose it and we just pile it up somewhere up there in a trunk. Well, these are the reasons or the issues which Cyomics like to address. At Cyomics, we have a huge community of experts and students from all over the world with various levels of experience and backgrounds, whom you can think of as your friend, philosopher, guide, whom you can talk to. There's no need to be shy, because if you join our community, every day you will find someone posting some questions which might be not very big in a matter of issue, but it's definitely treated with respect by every member of the Cyomics Club, because we have been there. We all have been there. We have all started from level zero. So it's like that particular friend whom you have always been hoping for to arrive in your life, with whom you can talk, with whom you can discuss, get advice, and move on with your research life. Also at Pine Biotech, just as we heard from all the experts today, we have great scientists with loads of experience in academia and industry who are there to guide you and who can be your mentors for making your research project actually take shape. And finally, when you work in a group, when you see others progressing, you also develop that motivation to get to the table, to power on your laptop and to get started with your project, to be at par with everyone in that particular community. So that healthy competition atmosphere motivates each one of us to get the best out of ourselves. So here at Cyomics, we would like to help you, first of all, to get a very good research idea or a research question, which is at par with what's happening all around the world right now, which you can get by attending several guest lectures, which would be absolutely free of cost, and they would be twice every month. So you get to attend their lectures, listen to them, get their advices, learn about various opportunities, about research going on around the world, what's the cutting edge situation right now in one particular domain, what's the skill sets required to start your own research project. Then you would get great mentors at Pine Biotech who can help you out with refining your question, with refining your idea, and also helping you out with the various skills or the tools that you need to perform this particular research, the methods. Finally, you can obviously talk to other peers who are part of this club, discuss problems, clarify your doubts, talk about certain errors that you might be coming across in your particular research project, or talk about new challenges which you can solve together. So that's a place where it happens, where you get new ideas. And finally, you would also get access to various training programs which would be launched by Pine Biotech in the near future, which you can get enrolled in to develop new skill sets that are very much prominent in today's world. So, briefly, I would like to talk about what are the various areas of interests to us, the club members, as well as to our mentors. Well, for example, the buzzword cancer, we are interested in learning how bioinformatics is changing the way we treat cancer, the way cancer research is progressing, and bioinformatics is at the core of those sort of research. So, that is definitely our number one interest. We are also very much interested in looking at various metabolic diseases like diabetes, and also we are interested in knowing and learning the role of bioinformatics in treating infectious diseases like COVID-19 and ultimately vaccine development. Because as you might know, the rapid development of vaccines all around the world was facilitated with the help of bioinformatics, especially. And there's lots more that's going on and that interests us. Many of them are very new to this domain of bioinformatics and omics. For example, personalized medicine, although not so new, but it's something which we are beginning to get a grasp of at a very young level of our career. So that's really another exciting domain which we would love to learn more from the guests and from the experts and the mentors that we have. Cheminformatics, that is a use of bioinformatics to develop drug targets and drug candidates with the help of molecular docking and other tools. That's also something which really interests us. Then comes my favorite area of research, which is astrobiology, which specifically is looking for molecular signatures or biological signatures throughout the universe, not only limited by the conditions on Earth, but also beyond. And not only that, but also to analyze the effects of space flight, the hazardous environment on the molecular level of the astronauts, and to actually leverage bioinformatics to understand what's going on in their bodies, in the cells, and to use that knowledge to transform the healthcare system on Earth itself. There are numerous examples of how that is being done, but we will talk about that maybe on one of the Cyomics club sessions. So definitely do consider joining the club. Then there's agrobiotech and much, much more that's going on. But the most exciting part, which you would get exposed to by being a part of Cyomics Club and also by being in close contact with Pine Biotech, is something which is known as faculty-led projects. So Pine Biotech has a lot of collaborations going on with universities and other organizations all around the world. And every university or organization have a big number of faculties who are interested in one particular domain of their research. They have been working on that domain for years. They have done their PhDs, their postdocs, and many years of research in those particular areas. Now, what you get as a benefit by being a part of this community is sometimes it might so happen that you do not have a research idea or a question of your own yet, because you are starting out right now. You are in the first or the second year or the third year of your undergrad, and you're really getting exposed to bioinformatics for the first time. That's pretty normal. I have been at that place myself, even. So, what's the best way to get started at that particular level is to get or to join a team of some great faculty all around the world who are working on their projects, and you just get to stand with them and look at their projects, look at the methods that they're using, look at how they're addressing the various challenges, how they're shaping their question, and you get to learn a lot by doing that. Now, as you might understand, it's often not possible to contact those faculties, to go to their organization, to go to their places. The only time you can do that is when you go for a research internship or something like that. And that's also for a short period of time, like 3 months. But the major advantage that you get by being a part of Cyomics and also by staying in close contact with Pine Biotech is you get exposed to such faculty-led project opportunities all throughout the world, not only in your country, but also beyond. Now you can participate in their research teams, get to work closely with them, attend team meetings, get to know the ways and the strategies they are developing to address the challenges and the way they are formulating their research project. And I find this particular opportunity really exciting because it can transform your entire career of bioinformatics research in the future. So, how would specifically our mentors and the courses which Ilya showed you in the beginning of the webinar that are already listed in the portal help you with your research project and to really get started with your research journey? Well, they would help you to put together all the pieces of the puzzle. To do a research is a big puzzle because there are various pieces like literature review, finding a good data set, finding a or defining a good research question, then bringing together all the required methods, interpreting the results, and coming up with a conclusion. These are all pieces of a puzzle. But if they can be put together in a logical way, they would look a beautiful picture like we see in a jigsaw puzzle. So that expertise, that guidance to do that will be provided by our mentors and all the asynchronous courses that you can go through yourself and also give you a direction in which you can head in the future, where you could get a master degree or a PhD or maybe a job opportunity in the future. All of this guidance and these information would be provided by our mentors, and you would get that if you are a part of the Cyomics Students Club. Now, this model of whatever I said has been tested and proven by various students, but today I would like to share my experience or my journey in brief, that how I benefited personally from this sort of model. So how I started learning bioinformatics as a young student who's doing his undergrad and knows nothing about bioinformatics, heard the term for the first time literally, and ending up becoming a spaceomics researcher, which is completely a niche domain, which is practically very new and very few people are working in this particular domain. So as you might have guessed, I was also a research intern at Pine Biotech. That's where I got baptized in bioinformatics, got to know about the various tools, got to know about what is data science in biology, how a curated data set looks like, how we can use the various pipelines, the various tools, interpret the results, formulate a research question, give shape to a research project. Everything I learned here. So you can see that was my certificate of the internship that I did. And here are some other certificates that I earned by going through the various tutorials out there in the Pine Biotech portal. So this is a place where I got all the necessary information to begin with. And then one fine day, I was looking through television and various other blogs. So as you might have already known, you guys have also come across various shows and such materials in Discovery or any other channels that astronauts face a lot of problems when they embark on a space mission, when they go to space. And that's all because of microgravity, radiation, isolation, etc., like bone and muscle density loss, physical strain, cancer, gut microbiome changes, etc. But that's not where I stopped. I took one step further to translate this information or this need for looking into this particular domain to convert it into my research interest and to convert it into my research project. So that's where, again, I got connected with Pine Biotech. I approached them with my idea, and they were very happy to welcome this idea. Although it was new and a very niche domain, they helped me all throughout the journey. The excellent mentors at Pine Biotech helped me at every stage, helped me address every challenge that I faced. And finally, I could devise some result out of the research question that I had, which gets aligned to NASA's human research program, that is basically to identify and to address the challenges faced by human beings inside the space conditions. So as you might see, some of those research are already in a preprint condition right now, and I'm hoping that they would get published. But that's not where I stop, because this is just the beginning. Now I am looking towards other data sets, more complex data sets, more complex organisms to look into and more complex pathways and biological processes which are out there to be analyzed. These data sets are available in the public domain. What they require is the proper interest, the proper motivation, and the proper guidance to analyze and get something very meaningful and outful, like useful from those particular data sets. And that's how being a part of this rich community helps you. That's how much the being a part of the community can elevate your ideologies and your experiences. And as you might have already guessed, this is where everybody of us once have been, the stage where we get an idea. And this is where we want to be. We want to be successful. We want to make an impact. We want to announce ourselves into this domain that, yeah, we have also done something. But there's a big gap between those two areas. And that's the gap which Cyomics loves to bridge. And that's our vision, to provide you with the necessary amount of guidance, the necessary amount of resources, information, and tools to go from here to here and make an impact into the world. So in a nutshell, the agenda of Cyomics looks like this. We would help you to learn about the current research standards and challenges present out there in various domains of interests all throughout. We would also help you and provide access to various new skills, and also our mentors would help you define impactful research questions. And finally, our community would also help you to interpret results, publish in various journals, or present it in some great conferences all throughout. How do we aim to provide you the information? Well, as I said, by inviting guests who have lots of experience from all over the world in various domains for a webinar, which you can attend for free. And here's what the timeline, which is quite tentative, but still is in our action of plan. So in November, we have the theme of cancer. So we will be having two great speakers talk about cancer, their research opportunities. In December, we would have infectious diseases. In January, we will talk about astrobiology. In February, metagenomics, microbiome. And March, personalized medicine, and so on and so forth. It will go on and on without stop. And every month, there would be a new theme, a new particular domain of science and bioinformatics, which would be addressed, talked about, and all of you are welcome to be a part of it. So if all of this sounds exciting and good to you, do consider joining Cyomics today. It's absolutely free of cost. We do not charge anything to be a part of Cyomics and to get access to the community as well as the various guest lectures. So you can join us through WhatsApp, so through Telegram or Slack channels. The details will be shared. You can also find all those details on social media platforms of Fine Biotech or my social media platform. So if you want to stay in touch with me, get in touch with me via LinkedIn or Instagram or Twitter. And I would love to talk to you. And if you guys have any questions, feel free to put them in the chat. I would love to address them. And finally, hope to see you in the next Cyomics sessions. Good luck with your research journey, and I wish all of you all the best. So right now, I would pass it on to Sonalika to take the session further. Thank you so much for listening to me today.
Thank you so much, Shubhroji, for sharing with us about the Cyomics Club. I'm sure the club is doing some amazing work. And I've also shared the link of the Slack group with you all. So this is what the Slack group looks like. And if you have registered yourself on the program page, then you are going to get the invitation to be a part of the Slack channel automatically. So all you need to do is just click on the link and you will be redirected to the Slack channel. Also, we have a Skype group, the details of which has already been shared in the chat box. So now I'll be just taking you all with the registration process, and also many of you asked about these scholarships. So let's now discuss how you proceed further to enroll yourself for this very exciting research fellowship program and get started with your research project. So once you visit this research fellowship program page, you are going to get all the details that you are supposed to know for the program. And today we discussed these 3 steps in greater detail. That is, you are going to get ready through training and then find an impactful research topic and present your analysis for expert feedback. The three major outcomes of the research fellowship program could be either you could come up with a literature review or present your research work in a poster presentation competition maybe. And the third one is definitely publishing your research work in reputed journals. And here on the program page itself, you will get the details of all the mentors. You heard from some of the mentors today. And also you could register yourself in the form here, which is mentioned in the program page. And once you register yourself, you are going to get all the details about how you can further enroll yourself for the program and how to get started. Now we do understand the need to make this program reasonable for students, along with the basic requirement of sustaining the program altogether. So we have designed this new program page for the computational biology internship program or the fellowship program. So don't get confused between these two terms. At many places for the term internship, fellowship word is being used. So you could find the basic structure of the research fellowship program here on the page itself. How do we proceed further? So first of all, you all would be supposed to go through a course material and then come up with your research proposal, submit your research question, and then get started with your research project. And how do we meet? We are going to meet twice every week to take up your doubts and see your progress, and also suggest you or recommend you the further steps that should be followed to carry out your project. And you can go through the structure here on the program page, which is very well defined. And to join the research fellowship program, you can take up the license here on the page itself. So we have three licenses. The first one is beginners, the second one is intermediate, and the third one is advanced. Now, these three levels are based on the different types of features it includes. So the beginner level does not include the one-on-one mentorship. That is that you could take up your doubts in group sessions. And also many times participants are not able to take up their doubts in a group session, maybe because they're not able to clarify their doubts clearly, or they do require a one-on-one meeting with the mentor. So that could be taken up in the intermediate license. And then for the advanced license, we also have the technical support that is being provided along with one-on-one mentorship. And also we have different types of license on the T-Bioinfo platform that Ilya showed you all. So there is a specialization license, an educational license, and a research license. Now this license has different provisions that it's going to provide you to work with your data. In the educational license, you would be able to use the big data from the various big data repositories, upload the data and work on it. And in a research license, if you are generating your own data, or you are from a lab or a PhD student maybe, then you can upload your own data using an FTP account and carry out the analysis. So this is the page where you can just enroll yourself using taking up any of the license that you would like to take up and enroll. And then on this page itself, you will also get to know about the various courses and sample projects that you can go through. Now, all the courses that Ilya also showed you, as a part of the research fellowship program, all the courses and the example projects would be unlocked for you all. So everything here under the courses tab, you can go through and learn from. Now, there is one more aspect that I would like to share with you all, that is the scholarship application. So we do have a monthly module of license that you all can take up and be a part of the program. So the module that I just showed you, that is a monthly module. That is, you have to take up this monthly license and carry out your research work. But if you would like to take up the license for a 3 months research fellowship program or a 6 months program altogether, then you are eligible to take up or apply for a scholarship application as well. And this is the scholarship application form that I will be just sharing in the chat box. Once you fill this scholarship form, we will review your application and get back to you. to tell you how much scholarship you're going to get for the research fellowship program, and that's the way you can get yourself enrolled. Now, I would like to leave the stage open for everyone to take the doubts. Please put in the doubts in the chat section, or you can also raise your hand and unmute yourself to take up your doubt.
Sonalika, I just wanted to clarify on a few things that were just mentioned. And hello, everyone. Myself, Mohit Mazumdar. I'm one of the co-mentors uh, uh, and representatives who are working on collaborations. Today, I wanted to discuss few things with all of you. So we have some of the exciting collaborations that we recently did, signing some of the MOUs with some of the university departments who are involved in research, some of the departments who are involved in training, and also industry where we have people from different pharma industry and the biotechnology industry to help students guide through. So about the licensing, which Sonalika was mentioning, it's actually a subscription. So when you're taking a monthly subscription, you're paying monthly. These can be done in 2 months, 3 months, and 6 months. The structure works for different models. So if you are thinking about taking a 2 month research fellowship program, you can go for either a beginner subscription or a intermediate subscription or an advanced subscription. So obviously these are different subscription levels that are available for you to take by monthly if you choose to, take the mentorship and take all the resources or the premium resources and develop a project. But obviously you can start from a very beginner's level that would not require you to take any license, not require you to pay anything. Just join the Omics Logic portal, and then you can also take help from Cyomics group, as mentioned by Shubhrajit, that would help you develop such research question. And then you can take the decision to go for big data analysis. So one of the reasons that there is a subscription to this program is because that we are actually providing you with a platform to do big data analysis. And that platform itself would require us to, give you a lot of space on cloud, as well as access to all the resources that we have developed. So that's why there's a subscription. And coming back to the question that Crystal has, that she's saying, I'm Brazilian and have no money. So that's perfectly fine. We have different types of scholarships that are available for different kind of responsibilities. So Crystal, we also have a research campus ambassador program, which you can join. And one of the basic objectives of that program is that you bring awareness about bioinformatics, about omics logic training in your area. And we kind of get that in a form of a report. And then we provide you a full scholarship to do this program for 3 months or 6 months. So that is also a way to do this program and get supported. But that would require you to give a lot of time and effort and learning and applying all of this. So the best way to develop a project is obviously to work directly with the mentor, do the research webinars. Come and join the research webinars weekly and discuss your problem. So we are available 24/7. And then there are like online courses and the portal that you just saw, which is available 24/7. So all of this actually combines to become a Omics Logic program or a research fellowship program. And I just wanted to quickly share with you about some of the university programs that we have. So we have collaborated with Amity University in Jaipur and in Noida. And if you are a student there, you can reach out to us and you can let us know, and we will be able to help you with registration relevant to the university. So just to tell you about the 3 different collaborations and the collaboration with industry. So we recently, just a second. So we recently collaborated with Amity University Jaipur, and there's a reason somehow I'm not being able to share the full screen. And we discussed about different multi-omics courses and integration of, integration of our programs into their internships. So students will be joining from MIT University Jaipur today. Over 130 people joined, and we discussed about the entire research fellowship program and development of projects in group. So one of the way to make it sustainable is to also work on group project or collaborative project. And all of the things that we have discussed today, and all of the things that we've been able to achieve, is because of the collaborations. Is because of that we are working as a team. So one of the biggest aspect of this kind of training and this kind of community is about collaboration, is about understanding each other, is about doing these tasks by dividing the tasks and by showcasing our own expertise in different areas. So similarly, we have a collaboration done with CGC Landron, Chandigarh. And then we have launched the research fellowship program for the students there. And there also we are working with Department of Biotechnology, Microbiology, and pharmacy to help students develop research projects in those areas and work through these different resources. And that all starts from the basic training. That all starts from the going through those basics that has been reiterated over and over. And then we do have another collaboration with MIT University Noida, which turned out to be like a historic kind of a collaboration because it involved several departments. So we are talking about at least 6 to 8 departments within MIT University Uttar Pradesh, and then defining of those research questions during that several meetings that we had. So we had brainstorming sessions where we discussed that how this can be integrated, how the expertise from faculty can be integrated into a program where we can help students, giving them a holistic understanding of how to work on or define a research project, and then work with a faculty-led research project, as Shubhrajit was telling in his presentation. So these are some of the opportunities that are there with universities and those universities who have reached out to us. And obviously, we are working on different type of projects within those universities. Along with that, one major collaboration that I would wanted to, showcase to you all is the collaboration with industry, which is with FABA Academy, which we recently did. And in this meeting, we discussed about how we can help, how industry can help in recruitment, how industry can help in giving job opportunities. And we met with people here, as you can see on the screen, that are leaders in industry, leaders in biotechnology. They are represented in big pharma companies, and they're also working with AI and drug discovery. So one of the objective of this collaboration is to be able to develop resources in data science and AI in healthcare. And I'll be working very closely with a group of people here, developing technologies and resources for students, and also giving students an opportunity to work on projects related to AI in drug discovery, where we are thinking about small molecule screening, in using information from protein folding, structure prediction using AlphaFold, that you must have heard about. That was recently came out and surprised the structural biology world, and not only the structural biology, but maybe the entire world. So another application, data science and AI in clinical trials. So this is a huge also when it comes to AI-based drug discovery. So what we are trying to do, and what we're trying to do in this program, would be helping you decide on some of the important research areas first, and then go to another step to work and develop the research project. So if you are not sure like which research area that you are more interested about, then you can go about the intermediate training after doing the basics. That would tell you about these different research areas such as precision medicine, population health, agriculture and biotechnology, Spaceomics that we discuss in length. So you'll be able to work on 21st century problems, and I'll be available to help you as a mentor. So I have expertise and over 40 publications in different research, addressing different research questions using bioinformatics. So please feel free to reach out to me and reach out to our team, and we'll be able to help you go through and complete this journey. And you already saw the subscription levels for the research fellowship program. You know about the Cyomics program, And it will be great if you also can become the ambassador of these programs and help others who can be a good fit for this. So with that, I would like to conclude today's session. And it was really, really great to see you all. And I'd really like to thank Dr. Raghavendran, Dr. Harpreet, Ilya, and Sonalik and Subrahjit for giving us wonderful lectures throughout this way. And we will be looking forward to meet you next week. So next week also we have a wonderful session coming up, and we'll be emailing you about that on different omics logic programs that we just discussed. So we'll be talking about infectious diseases in much more detail. We'll talk about metagenomics. We'll talk about genomics. We will talk about transcriptomics. And these specialization programs are how they are helpful in developing and in understanding and finding those research questions. So thank you.
Thank you so much, Dr. Mohit. Thank you so much, everyone, for being a part of the webinar and making it such a wonderful event today. And if you have not registered yourself, then do register so that you get the links to all the groups that you can join and stay updated about the program. Thank you, everyone. We'll be meeting soon. Bye-bye.